In terms of having a model for how skeletal muscle works, it's important to recognize that skeletal muscle will not contract until it's stimulated by an action potential arising from a motor neuron. So we have command signals for the central nervous system emerging through these alpha motor neurons, the spinal neurons. That neuronal stimulation is then transferred to the muscle through the neuromuscular junction, which is the topic of this session. The NMJ there is the synapse then between the motor neuron and the skeletal muscle cell, sometimes also called an end plate because of its structural organization. That will then lead to an action potential in the skeletal muscle fiber, and we'll talk in the next session then about excitation, contraction, or EC coupling. Each skeletal muscle fiber is a single cell, and each one has only one neuromuscular junction, and that arises around about its midpoint here. So here's the alpha motor neuron coming from the spinal cord, synapsing down here on muscle fibers. An individual neuromuscular junction then consists of small branched patches of bulb-shaped nerve endings called terminal boutons. And this diagram at the bottom here is a kind of a cutaway of a single bouton. If we completed it there, it'd be more sort of circular kind of a structure. Motor neurons branch as they activate a group of muscle fibers, and this branching here creates what's called a motor unit. In this example, there are only two branches, and so this motor unit only innervates two individual muscle fibers. Muscles that are subject to fine control, such as muscles of the hand, have many small motor units, whereas postural muscles, such as the lower back, would have fewer large motor units, where the axon here will branch many times to potentially dozens of individual muscle fibers. The neurotransmitter at the neuromuscular junction is acetylcholine, which is synthesized in the motor neuron terminals by combining acetyl coenzyme A with choline, and that occurs via the enzyme choline acetyltransferase. The acetylcholine then is stored within vesicles within these terminal boutons here. The postsynaptic muscle cell membrane immediately opposite the presynaptic terminals has a high density of nicotinic acetylcholine receptors. These are ionotropic receptors that function as non-selective sodium and potassium channels. When they're opened, they cause skeletal muscle cell membrane to depolarize because the combined equilibrium potential for sodium and potassium, E cation, is approximately 0 millivolts. If we were to take a single presynaptic vesicle and release the acetylcholine in just one vesicle, that's an amount of acetylcholine that is sometimes referred to as a quantum. And if we were to look at the electrical effect on the muscle cell membrane of a single quantum being released, we would measure what's called a miniature end plate potential. When many quanta are released at the same time, these miniature potentials summate to produce a full end plate potential. These are examples of excitatory postsynaptic potentials. Under the normal conditions, a single motor nerve impulse will normally produce enough quantal release to bring our end plate potential through threshold for an action potential to be generated in the muscle cell membrane. Acetylcholine within the synaptic cleft is rapidly broken down to choline and acetic acid, and that occurs through the action of the enzyme acetylcholinesterase. And this enzyme is anchored to the postsynaptic membrane. Choline would then be retaken up into the presynaptic nerve terminal to be reused and recycled for acetylcholine synthesis. A high-yield thing you should be aware of is that myasthenia gravis is an autoimmune condition in which antibodies are directed against the nicotinic acetylcholine receptor. The release of acetylcholine is normal, but the postsynaptic membrane is less responsive, and this results in muscle weakness. Myasthenia gravis can be treated with agents that inhibit acetylcholinesterase in the cleft. This prolongs the action of acetylcholine at the neuromuscular junction and increases the probability that action potentials in the muscle can be achieved. Another form of myasthenia, known as Lambert-Eaton syndrome, can occur. This time the target happens to be calcium channels in the terminal bouton presynaptic membrane, and the defect in this case is the release of acetylcholine. In the next session, we'll pick up on the action potential in the muscle membrane and describe how this results in muscle contraction.